So hello, everyone. Welcome back to our mini conferences. Very excited to be kicking these off again with our newest CanDIGger who has quite literally hit the ground sprinting since joining our team. So Francis will be presenting for us today on the CanDIG variant search. So Francis, whenever you're ready.
Awesome. Thank you, Samantha, for the intro. As Sam just stated, I'm Francis Quinn. I just moved over to the CanDIG team circa, I think, like November of last year, or maybe it was December of last year. Moving over from the cards team. And today I'm just going to present the current work that's being done on the CanDIG team's variant search platform. So just I'm going to make sure that everyone's on the same page. I'll be introducing what genomic variants are, why we're interested in implementing this functionality into CanDIG, followed by telling you about what we currently have in regards to variant search, and finally just moving on to stuff that we are currently developing. So first off, just to make sure that everyone's on the same page, let's talk a little bit about genomic variants. Genomic variants are alterations to the genome, either like modifications to like a single nucleotide. We've got like genomic variants that are deletions of certain areas of your genome, insertions of new transcripts or other nucleotides into your genome, and a bunch of other things that are mostly just also insertions or deletions of a sort. And something. Usually in genomics, we talk about variants as compared to a genomic assembly. So there's a consortia of teams in the US, I believe they're mostly in, that have decided that, hey, this is what the human genome looks like right now. And they release these genomic assemblies with code names that are incredibly helpful, like GRCH38 or HG19. Basically, they're just code names for different consensus genomes that everyone's like, hey, this is what the human genome looks like right now. And then we can define genomic variants as like differences from that. In addition, though, in oncology and I'm sure in other diseases as well, we can also call genomic variants things that are different between, some part of the, some part of their tissue, which we might call like normal tissue, versus, disease tissue, like tumors. So there's kind of like two definitions of a variant in this case. As for why these genomic variants are so interesting, it's because these genomic variants, they're a pretty large focus of medical research. These genomic variants can, since they are alterations to the genome, they can be the main drivers behind cancer. But it's never quite so simple as like mutation A causes disease B. Sometimes you can sort of link together things like that, but mostly you need like multiple such mutations at the same time in order to have some effect on your individual. The research that I've helped with in the past is focused on the determination of things called genomic signatures, which are combinations of these sorts of genetic variants that can alter patient prognosis. So let's say, you have a disease, you come in with, let's say, prostate cancer. Can we identify, a certain number of variants or a certain pattern of variants that are associated with poorer patient prognosis, that is like poorer outcomes when, doing surgical resection, that is like removing the tumor? Or perhaps, are there a combination of genomic variants that are associated with like poorer outcomes after chemotherapy, or perhaps resistance to certain kinds of chemotherapy? So determining these genomic variants are, a very important part of modern day oncological research. The Terry Fox Institute has data from the Marathon of Hope Cancer Centers Network, which is one long acronym, MOHCCN, data that we need to make available to researchers across Canada. We know that their data adheres to some specific data model, which we then index and make available to authorized individuals. You obviously have to like obey ethics and privacy concerns, with your data. That's like part of what you sign off on when you release your data through these hospitals. So the goal of CanDIG, I realize almost everyone in this call is or has worked with CanDIG for a long time, so maybe the slide was a bit unnecessary, but CanDIG is a data discovery platform. That means that, like, rather than just being interested in just being like a store of information, what we're aiming to do is make data available to institutions and consortia so that they can use information about patients for good. We, in particular, are hoping to allow for discovery queries, such that researchers can find if there exist patients that fit their criteria, like certain types of cancers with certain genomic variants, if they're interested just in patients who display, like, I don't know, BRCA1 mutation, but like not in breast cancer. We can help answer those sorts of interesting questions to researchers. We also have to do these queries in such a way that hospitals have full control over their own data. They are always in full control over like who is able to see what, and just for their own like privacy concerns. And moreover, patients can ensure that their data is only being used according to the data release policies that they've signed off on. And I believe most of this was covered in a previous talk about authorization done by Daisy. So just as a quick overview of Beacon, which is done by consortium GA4GH, they allow us to do sort of discovery query by taking the omics data sets from private entities, from research entities, from hospitals, constructing their own indexes, and then afterwards a researcher can just ask for, hey, show me all variants in rare disease X. You send that off to the Beacon network, or in our case, the CanDIG network. Each of them, or each of the participating sites, responds with what they have, and then we aggregate this response and bring it back. And this is what's called federation. And on an even simpler scale, it just looks like, hey, do you have this particular base pair at this particular position? Yes or no? And across each individual person, across every single person that's inside of the CanDIG or Beacon network, it'll just aggregate these responses of like, yes, this patient, no to these patients, or like, yes, there are 5 patients that have a C at chromosome 13, et cetera, et cetera. The challenges in implementing our own, though, is that, like, obviously searching for like a particular data point across like a huge amount of data, because these are like petabytes of data across like all of these different institutions, is being able to do these searches in a performant manner, like being able to respond quickly with these discovery queries. In addition, we weren't totally clear, like, hey, when researchers are looking for variants, are they necessarily looking for variations from the genomic assembly, or are they looking for variants from tumor selection or from their normal sample? And this wasn't quite clear to us after talking with some of the researchers. Moreover, there was a question of zygosity, so I forgot to describe the zygosity, but you know how every human has 2 copies of every chromosome. Are we looking for complete heterozygosity, which is to say, like, are we looking for the variant to always be in one of the 2 chromosomes? Are we looking for how prevalent does this particular change, does C at this position have to be across your sample before we return it? And this, we could be excluding, or researchers might be interested in excluding certain like low zygosity counts if they're uncertain of like the quality of the data that they might be querying for. So now we're going to talk about the current implementation of variant search. Most of this backend was implemented by Daisy, and most of the front end was implemented by Courtney. Actually, I think yes. So let's talk a little bit about ingestion of data. So we receive these like, petabytes, terabytes, petabytes. I'm not entirely certain what the full scale is. These giant files, and obviously we can't just scan through petabytes of information in order to return, a particular query. So instead, also we can't just copy all this file because it would just double the amount of space that hospitals require in order to be able to facilitate CanDIG. So we process VCF files by indexing each of the VCF files, mapping similar headers across the files, and then we do something really interesting. We, for every single 10 kilobase pair bucket in the human genome, record which VCF files have an entry in that bucket, and we create a small kind of index for that. And store all of this into a SQLite database. So just as a quick UML diagram of what sorts of information that we store in these indexes, we store like the CanDIGs, which is like the assembly. Sorry. Okay. We store information about like the headers of each VCF file, the information about the variants that each VCF file contains, information about the sample, et cetera, from the VCF files. And just to illustrate kind of what this 10 kilobase pair binning looks like, suppose we have a region of the genome, and for this example case, we have two VCF files that we're interested in indexing. We've got patient A and patient B. And they have single nucleotide variants or single nucleotide polymorphisms in different regions of the genome. Here I've just sort of zoomed into, I forget what region this is. But yeah, in order to ingest this, we look at each bin, which is 10 kilobase pairs by default. We can adjust that as necessary. And we construct these indexes of like, hey, instead of storing the full data about like every single nucleotide variant inside of this, and there might be like multiple per bin, we instead just store like a binary yes, no for, hey, inside of this 10 kilobase pair bin, patient A has data in here, patient B does not. And no patients have data in this bin or in these three bins. Patient A has information in this bin over here, followed by another three, and then somewhere where patient B has information. By constructing these indexes, we can sort of drastically cut down on like how much information we need to store, and also make it a lot faster for researchers to then, find their data. We make it a lot easier for our backend in order to find out like, hey, which VCF files are relevant for a query. So speaking of queries, when you're doing a discovery query, you transform the search into buckets, just as we previously saw. We check which files contain an entry in each of these buckets. We run PySam to grab on the original VCF files, grab only the parts that are relevant, and then we process these files using the headers that we previously mapped to VCF files to obtain information. So let's say like a request comes in for variants in, I guess this was showing chromosome 17 at a specific 30 kilobase pair range. We transform this request into, a certain region of the genome. From the indexes, we know that only patient A has variants in this region. So then we can grab from the original VCF file, we can use PySam and grab out any of the variants that it has discovered in this region, combine that with header information to return the metadata on the original sample, and voila. Just as a quick overview of what like the sample, for what the headers might look like, it might contain information that's really relevant to researchers, like the quality of the calls. It might contain information like the genotype, which is very important, like the read depth, which is like how many things correspond. And as a little preview of what the HTS-GET protocol that what Daisy was working on returns, you get even more information about like the specific variants. So like, here's a single nucleotide polymorphism at this location. And like, the level of zygosity, for instance, et cetera, et cetera. Of course, I think a lot of our researchers won't just be like making these raw quals. They will instead go through the front end. So this is mostly Courtney's work. This is a Figma prototype of what the front end will eventually look like. Just as mentioned before, there's federations. So you've got like a certain number of nodes, or when you make a discovery query, you are querying across like a certain number of nodes, across a certain number of hospitals, across a certain number of provinces. And as for the actual, what the federated search looks like, currently it looks like this. So let me go through each of these by parts. So you would enter in at the top either a region of the genome by start and end position, or via gene name, which is an older version. You can also do like an autofill in gene search over there, using NCABI refseq select genes to convert the gene names into like an actual position for the backend to use. You can then obtain VCF data and also metadata about these patients, which I'll show shortly. You can filter the results by editing the sidebar. So this is sort of similar to how other genomic sites allow you to filter data, like CBioPortal. Basically, there's an adage in UI design that people spend a lot more time on every site but yours. And so if you make your filtering look like how bioinformaticians are used to filtering work, then we can get the best buy-in. So that's sort of why we did this. We made this look like how CBioPortal does it. You just enter in which nodes you're interested in, the cohorts, the primary sites, treatment, etc. Return these to the backend, and out comes like a filtered list to your front end. If the researcher does not have the credentials necessary to view these patients, instead you get the sort of counts of results that you would have gotten. And if the counts of results are too low, then obviously we have to do a little bit of censoring, because otherwise it becomes possible to identify individuals just by curating your search, which would be a leakage of patient information, which is bad. Since our backend does support like the CA4GH HTS-GET protocol, we also have the bonus feature that we are already interoperable with software that can take in data from HTS-GET servers, such as IGV viewers online. And I think this is like a feature that quite a few researchers have expressed interest in. Also, thanks to Courtney, we have customizable graphs of the patient distribution in your return types. I don't think we have yet mocked like how you can edit these graphs, but that's something that we're currently working on. Another thing that we're currently working on is trying to figure out how we can display per individual results. So previously, you would select a particular patient and we would just return a giant JSON file. Now we're using something called pivot table, which, just zoom in. This pivot table allows you to sort of see at a glance, you would be able to like enter in things that you are interested in, and it would show you, hey, there's one sample who is from the oropharynx of a woman who is currently undergoing chemotherapy using oxaliplatin, et cetera, et cetera. This is also still, I think some of the feedback mentioned that this pivot table is still a little confusing. So we're currently trying to, figure out better ways to display this information. So for our future plans, we are still working on the pivot table. I mean, I like the pivot table. I think that's nice. Still trying to figure out the best way of showing all the details for a single patient. We still need to do benchmarking tests of our implementation to other Beacon implementations, such as Bento Gohan, and finally determine if there are any other use cases that researchers would like to use our service for that we can implement. So the takeaways for this presentation are just that genomic variants or variation of the genome that could be of interest, and CanDIG allows researchers to find genomic variants of interest in a performant manner while giving social full control over the system. Thank you so much for listening. Are there any questions?
That was great. Thank you very much, Francis.
Yeah, I just want to shout out to Frances, who, as Sam mentioned, joined our team not very long ago, hit the ground running, and is now giving presentations on a part of the architecture he hasn't actually worked that much with. So yeah,
and
yeah,
is very poorly defined on every single level.
Thank you.
So
Well, I
very,
mean,
very nicely done, Frances. Yeah.
huge shout outs to everybody that helped with the presentation, and also especially Daisy, who I think I interrupted in the middle of something last night when I was desperately trying to get answers to some questions that I hadn't thought of yet. So thank you to everyone involved.
Yeah, I just wanted to say it was a
Questions?
really nice presentation,
Yeah.
Francis. Yeah.
Thank you.